So, ladies and gentlemen, I think we'll begin. I understand there are probably some sessions overrunning slightly, and folks will come in, but I think we'll begin. So I'm Mark Ferguson. I'm the Director General of Science Foundation Ireland, and it's my great pleasure to be able to introduce Dr. Craig Ventnor this evening. So Craig is widely regarded as one of the leading scientists of the 21st century, an entrepreneur, and a really interesting person. He's best known for being one of the first people to sequence the human genome and for creating the first cell with a synthetic genome. And he began the race to sequence the human genome when he unexpectedly announced to a room full of genomic researchers that they could just quit now, thank you, because his company would complete the task quicker, faster, cheaper than anyone else. People who like Craig say that he rarely filters his thoughts and usually shoots from the hip. So John Craig Ventnor was born on the 14th of October 1946 in Salt Lake City, the youngest son of an excommunicated Mormon who drank too much, smoked too much, and died at the age of 59. The family then moved to a working-class suburb of San Francisco and lived in a house close to the train tracks. Ventnor enjoyed playing chicken with the trains and surfing the waves. According to his biography, A Life Decoded, he was not an engaged student, having C's and D's in his 8th grade report cards. Ventnor was drafted and enlisted in the US Navy for the Vietnam War, a war that he opposed, and was trained as a medical corpsman and shipped to Da Nang Hospital. There, he got introduced to medicine in the toughest way possible and decided to study it. After finishing his tour, which included 2 stints in the brig for disobeying orders, Ventnor went to the University of California, San Diego with the intention of becoming a doctor. He was deflected from that path, fortunately for all of us, by a class with Gordon Sato and a project with Nate Kaplan, and he decided to become a biomedical researcher. He breezed through undergraduate and graduate school at UCSD, became an associate and then full professor at the State University of New York at Buffalo, and then joined the NIH in 1984. When at the NIH, Ventnor learned of a technique for rapidly identifying all of the mRNAs present in a cell and began to use it to study human brain genes. The short cDNA sequence fragments discovered by this method, called expression sequence tags, were the basis for shotgun sequencing, which Ventnor promoted, and this was the fastest and most effective way to get useful human genome data. The method was controversial. Some geneticists felt that it was not accurate enough for a genome as complicated and as large as that of humans. So somewhat frustrated with the slow pace of the public human genome project and unable to get funds from the public sources, Ventnor went to the private sector and funded Celera Genomics. The goal of the company was to sequence the entire human genome and release it into the public domain for non-commercial use in much less time and for much less cost than the public genome effort. The company planned to monetize their work by creating a value-added database of genomic data which could be subscribed to for a fee. The goal subsequently put pressure on the public genome project. It spurred several groups to accelerate and redouble their efforts to produce the full sequence. Now, although Celera was originally set to sequence the DNA from 5 humans, partway through the sequencing, Ventnor switched the samples for his own DNA sequence. In 2000, Ventnor and Francis Collins of the National Institute of Health and the US Public Genome Project jointly made the announcement of the sequencing and mapping of the human genome a full 3 years ahead of the expected end of the public program. Despite claims that the shotgun sequencing method was in some ways less accurate than the clone-by-clone method chosen by the public project, the technique became widely accepted by the community and is still de facto the standard today. After contributing to the Human Genome Project, Ventnor was promptly fired by Celera in 2002. And if you want to know why he was fired, read his biography, because I'm not going to tell you. But next up was the Global Ocean Sampling Expedition, which he announced in 2004. This was an ocean exploration program with the goal of assessing the genetic diversity in marine microbial communities to understand some of nature's most fundamental processes. The project, which used Craig Ventnor's personal yacht, started in Halifax, Canada, circumnavigated the globe, and returned to the US in January 2006. Craig Ventnor is currently the president of the J. Craig Ventnor Institute, which conducts research in synthetic biology, and in June 2005, he co-founded Synthetic Genomics, a firm dedicated to using modified microorganisms to produce clean fuels and biochemicals. And in July 2009, for example, ExxonMobil announced a $600 million collaboration with Synthetic Genomics to research and develop next-generation biofuels. Ventnor has created an artificial life form, the first person to do so, and this line of research holds much promise for synthetic biology and for much of what we currently do by other methods. In May 2010, a team of scientists led by Ventnor became the first to successfully create what is called synthetic life. This was done by synthesizing a very long DNA molecule containing an entire bacterial genome and introducing this into another cell. The single-cell organism also contained 4 watermarks written into its DNA to identify it as a synthetic organism. Those watermarks included a code table for the entire alphabet with punctuation, the names of the 46 contributing scientists, 3 quotations, one of which comes from James Joyce, and the web address for the cell, since it was the first cell essentially created from a computer. On the 4th of September 2007, a team led by Sam Levy published the first complete 6 billion letter genome of an individual human, Ventnor's own DNA sequence. So it is a very rare honor to be able to introduce a keynote speaker whose DNA you can analyze before the introduction. So if you launch the ensemble and you look at Craig Ventnor's DNA, what do you find? Well, you find a genetic predisposition to wet earwax. You also find an increased risk of antisocial behavior, which I fear I may be on the receiving end at the end of this introduction. And you also find a genetic predisposition to Alzheimer's and cardiovascular diseases. Ventnor's DNA is out there, and everyone can have a look at it, and I encourage you to do so. Craig has been the subject of several books, TV documentaries, and magazine articles. He was listed in Time magazine 2007 and 2008 on the list of the 100 most influential people in the world, and in the 2010 New Statement list of the world's most influential figures. He's published very widely and is very widely cited. His autobiography, A Life Decoded, My Genome, My Life, was published in 2007. So it is now my great pleasure to introduce Dr. Craig Ventnor, one of the world's leading scientists, an entrepreneur, and a really interesting person, to deliver the keynote address, From Reading to Writing the Genetic Code. Craig.
Well, thank you for a most novel introduction. It was mostly correct. I'll correct some of the errors along the way. My father was definitely not an alcoholic, but he was definitely excommunicated from the Mormon Church. It was coffee and cigarettes, not alcohol and cigarettes, a minor difference in some circles. But I'm delighted to be here and for my second lecture while in Dublin. And let me start by introducing my organizations. The Ventnor Institute is now on both coasts in the US. It started out in Rockville, Maryland, near the National Institutes of Health. And a few years ago, we also started a branch in La Jolla, California, just outside of San Diego. Also, Synthetic Genomics, as you heard about, is also in La Jolla, California. And we're building on the University of California San Diego campus the first carbon neutral research building. So it's exciting to be speaking, and I think this is the first carbon neutral convention center in the world. So it's a different type of energy production. Being California instead of Dublin, we can use sunlight for energy. And we'll be making extensive use of solar energy, but also heat exchange, water-cooled computers, et cetera. So this will be finished in the fall of next year, which will actually be the 21st anniversary of my institution. Now, as I mentioned the other night with the Schrödinger lecture, we've been reading the genetic code now for quite a while, and when we read the genetic code, I view that we're converting an analog information molecule into ones and zeros in the computer. I call this digitizing biology, and now the computers are filling up with vast amounts of digital biological information, but we're still in the infancy of this, and we probably have only a tiny fraction of a % of what will be accumulated in the next few decades. So let me talk first about reading the genetic code and digitizing biology. The EST sequences that we invented and the method gave us so many random cDNA sequences that we had to develop some new mathematical tools to be able to compile them. And we were looking how to go back into genomics to do whole genomes, and we thought we had some really novel algorithms for assembling genomes. So when my friend and colleague now, Ham Smith, who is at the Venter Institute, suggested we sequenced his favorite organism, Haemophilus influenzae, we took on the challenge. And this ended up being the first genome of a living organism, and was published in Science in 1995. It was only 5 years after this, obviously scaling up at a tremendous level, lots of different pathogens and other organisms during that 5 years, and the human genome was preceded with our test genome of the Drosophila genome, working with Jerry Rubin. And I think people were absolutely stunned that the algorithms, the mathematics, the computing were in fact the solution to genomics. And as you heard in the introduction, the method of whole genome shotgun sequencing is now the global standard for sequencing genomes. This is the team at the White House in the year 2000. The young man on the far left is Mark Adams, who was in charge of all the sequencing. The tall gentleman with white hair is Ham Smith, who discovered restriction enzymes in 1970 and shared the Nobel Prize in 1978. The guy next to me is Gene Myers, who led the team to develop all the algorithms. And when we started Celera, he had a team of mathematicians and coders that had to write 500,000 lines of computer code in a matter of a few months so we could test that on the Drosophila sequence. Labs used to be massive, as many of you know. At Celera, we actually had several giant rooms with 300 applied biosystems DNA sequencers working around the clock for 9 months to sequence the human genome. And on the computing side, we actually had to build the third largest civilian computer in the world in 1999 to be able to deal with and assemble this data. This computer was 1.5 teraflops. This is the same team celebrating the 10th anniversary of the human genome. And this picture proves you look better in a tuxedo. But everybody's still alive and well. But those giant rooms now were reduced to something smaller than this podium. And even though 9 months seemed surprising to people that had planned on doing the genome over 15 years, we can now sequence a human genome in about 2 hours. So it's moving into the realm of clinical diagnostic potential. Everybody measures the cost of sequencing. I somewhat facetiously say you can sequence a genome for $1,000, but you need $10,000 of compute time to do anything with it. So we're still not addressing the full picture, but the technology changes in a short period of time is stunning. Instead of a room about half the size for a 1.5 teraflop computer, now you can get teraflop boards to add to your PC. So fortunately, computing is still changing to keep pace with the changes in data generation. In 2007, as you heard, we continued sequencing. Actually, the paper that we published in 2000 was the compilation of 5 genomes, 3 women and 2 men. The 2 men were Ham Smith and myself, so it was not hijacked in just my genome. But this genome was just my genome because it was the first diploid genome, and to do that you have to have a single individual. And this changed our view of the genome. In 2000, people thought there was 1 genetic variation per 1,000 base pairs. All of a sudden, when we could have a complete diploid genome, we found that the variation was more like 1 to 3%. My 2 parental chromosomes differed by 0.5% from each other. So if we can compare my genome to any of your genomes, we're comparing 4 sets of chromosomes. So we're somewhere between 1 and 3% different between unrelated humans. And there's a lot more differences, as many people know now, than just single nucleotide variations. Vanessa Hayes at the Institute has been using genome sequencing to track human populations, and she did the first genomes in Africa, doing the Bushman genome, then doing Desmond Tutu's genome, who in fact joined us for the 10th anniversary of the human genome. And when we start to compare genomes, so the BGH in China did the first Han Chinese genome, comparing my genome to a Bushman genome to a Han Chinese, everything came out in this 1 to 3% of variation. But Vanessa's team, looking just within Africa, found even greater variation than comparing populations outside of Africa, and has now been working to narrow down to a part in southern Africa where it looks like the oldest genomes are. And this is obviously complicated because, as most people know now, we all evolved from populations that migrated out of Africa, but some of those populations also migrated back and intermixed as well. So it's not at all a simple story. Sorting out genomes into haploids is a very key part of the future of genetics. I mean, it's a question all of you have asked yourself about your relationship with your parents and your relationship with your children. What trait did I get from my mother? What traits did I get from my father? And you can't sort that out of just getting a bunch of genome sequence and having it all bundled together. So separating out into the parental chromosomes gives us very specific information about what we inherited from whom. And there's a variety of ways of doing this now, but just to show you how the technology is advancing, Roger Laskin at the Venture Institute developed single-cell sequencing. We can do a genome on a single bacteria or on a single human cell. Here is actually sorting out single sperm cells. We isolate those, and you can actually do a genome sequence on a single sperm cell. Sperm being haploid gives you a haplotype information. And what we worked out is there's approximately 1 crossover per human chromosome on average. So I always use the wrong metaphor and say if you sequence a handful of sperm, that you can very quickly sort out the complete haploid nature of a genome and compare it to parental chromosomes. There's mathematical approaches developing out of some of the newer longer range sequencing technologies, but they still need to be proven. So it turns out we have compound heterozygotes in genomics that, in fact, tell us a lot more about disease predictions and human variation. And this is just looking at the information coming out of the first diploid genome and looking at the number of non-synonymous SNPs, but then looking at actually complex compound heterozygotes actually predicting damaging properties and changes in genes. Some of these become very important, and there was a session previous to this on personal genomics. Some examples are shown on this slide. Obviously, Pfizer's drug, when they went to do a clinical trial on trying to treat lung cancer with their new drug, the trial failed. When it was found that there was a rare change in a gene that actually conferred properties where this new drug worked, while it's a small number of patients, 2 to 3% of the individuals with lung cancer, if you have lung cancer and you're one of those 2 to 3% of the cases, this drug seems to be quite effective, shrinking tumors in over 60% of the patients. So pharmaceutical companies are now changing their paradigm. To them, a blockbuster drug was something that generated billions of dollars in sales, and they did this by selling to a large number of people. Now the new model seems to be greatly jacking up the price to deal with a small number of people. So this new lung cancer treatment cost about $10,000 a month per treatment. Obviously, Herceptin and other drugs are coming up, and a new drug was just released to deal with one genetic variation in cystic fibrosis. So I think we're finally going to see genetics introduced into pharmaceutics and hopefully sooner into clinical trials instead of much later. We're not alone with our 20,000 some odd human genes and 100 trillion cells. We have twice as many bacterial cells associated with our body as we do human cells. And when Karen Nelson at my institute did the first shotgun sequencing of microbes from the human gut, that started what we now know as the human microbiome. And we have a large number of species associated with us. So our 20,000 or 22,000 human genes are supplemented by about 10 million additional microbial genes. And it doesn't matter how often you wash your hands, your skin is a very rich source of multiple species, and basically every part of human existence is very much affected by these microbes. NIH has been funding this work on a global basis now, and in May 2010 was a reference sequence of a very large number of individual genomes that were sequenced from the human microbiome set. But changes in association with human diseases are showing up in more and more diseases associated with our microbes. One of my favorites actually is most people think that alcohol damages your liver, which it can, but this is not a direct effect of alcohol on the liver. It's actually alcohol causes a certain microbial toxin to be released, which can damage the liver. So this might be the first city on the planet that might take my prescription. I suggest that we actually fortify alcohol with antibiotics so that you can kill the microbes and not damage your liver as you consume large amounts of alcohol. But this is not yet caught on in clinical practice. So what's the metabolic potential of all these genes? How do they affect us? And this new field of metabolomics actually measures all the chemicals in the blood, increasing our analysis of complexity. So our genes can produce on the order of 24 to 2,500 unique chemicals, or the protein products from those can do that. And these can be readily measured in the bloodstream, for example, after a meal. And what's found is about 500 chemicals circulating in our bloodstream. But only 60% of those are from human metabolism. 30% are from all the different species you consume in your diet. But 10%, or about 50 chemicals right now circulating through your brain, are bacterial metabolites of what you get from your diet and from the human chemicals. We don't know what these do. We don't know if these make you feel better, feel worse, make you sick, protect you. And so we're starting to get this very complex picture of our own biology. It's dependent on our own genetics. Our genetics determine in part which microbes are associated with us. Those microbes, plus our human genetics, plus what we consume and are exposed to in our environment, determines which chemicals are circulating in our bloodstream. So at least we have a chance. All this complexity makes me more optimistic that we have a chance to get to real answers now than when it was all a mystery. So sequencing is cheap and relatively cheap and plentiful. A lot of people talk about scaling up DNA sequencing. But other than tracking human populations and human variation, DNA sequencing on its own right now is not going to tell you very much. So if you look at my book, A Life Decoded, we can't tell much more about your genome today than 5 or so years ago when mine was sequenced. It's because there's very little data being generated by the genetics community of relevance on the scale of the human genome. And so what I've been advocating for is we need to learn how to develop a digital human phenotype. And without phenotypic information, genomic data is not of much use. But doing this is going to take a global effort because we have to categorize everything associated with us as humans, everything from our medical history, our physical descriptions, our chemical composition, disease history, family history, how your thought patterns work, et cetera. And we have to do this with a minimum. The starting point is 10,000 people, coupled with the human genome and the microbiome, to start to get a clear understanding of predictions for diseases. People are looking at the so-called disease genes, as we've seen with breast cancer, and people have backed off tremendously on how predictive they are. And very few genetic changes result in yes/no answers in the genome. So we have as many genes and gene functions protecting us from disease as we have genes that can cause disease. And if we don't look at the whole picture with a complete phenotype, we'll never get to meaningful answers. We've been using the same shotgun sequencing that we did for the human microbiome to look at the environment on a broader scale, and this is what started the Sorcerer II expedition. Many people think this is one of my greatest accomplishments because I found a way to sail around the world on my own boat and do science. And it was particularly rewarding. You can see the routes that we covered and circumnavigations plus additional trips up and down the US coast and then over to Europe. The first study was looking at the Sargasso Sea, where people thought there were very few microbes. In fact, they were numbered. SAR 11 was the 11th microbe discovered in the Sargasso Sea. So when we were looking for a place to test shotgun sequencing, everybody thought that would be a very simple test because there were so few microbes. We could test to see whether the mathematics of assembly could deal with these simple environments. We just filtered 200 liters of seawater and did a shotgun sequencing. We stopped sequencing after we discovered a little over 1 million new genes and found out there was tremendous complexity. We extended this and started the expedition. A couple years later, PLOS Biology published a special issue dealing with this information. And more recently, 2 years ago, we sampled in the Baltic Sea, which is a tremendous gradient of fresh down to salt water. And a year ago, sampled the Mediterranean Sea, and this was very tough to find volunteers to sail around Italy and Greece and up into the Black Sea, taking extensive samples along the way. So all this data is being compiled. It's basically plugged up some of the biggest computers in the world because of its vastness. And this is the type of data we see. This is a simple graphical tool that Doug Rush at the Institute developed, where you can put a sequenced microbial genome across the top as a reference, and each of these little tiny bars represents about 900 base pairs of DNA sequence from a read, and you can compare its degree of sequence identity to the reference genome. SAR11 was thought to be a single microbe. It turns out that instead of having the 1 to 3% variation in sequence, we get variation up to 50%. But because the average microbial gene is around 900 base pairs, when you randomly sequence 900 base pairs, you statistically cover 1.4 genes per sequence. So we can get the nearest neighbor off of each one. And so we know that basically all these different genomes here have largely the same genes in the same order. We see big gaps like you see there in the case of this particular genome because there was a viral insertion in the reference genome. So you see some changes, but these are largely the same genomes with the same genes in the same order, but up to 50% variation. All mammals differ from each other by about 10% sequence variation. So this is tremendous variation. In fact, my view is this variation is caused by two things. It's caused by UV light and by oxygen. And I'll show you why we think that in a minute. We can actually assemble just from fragments around genomes and from cells that have never been seen before, and we can start to compile all this data. So what people thought were individual organisms turn out to be entire taxa. And so the more data we collect from around the globe, the clearer the picture starts to get instead of more complicated. SAR11, a small photosynthetic organism, thus far seems to be the most abundant taxa in the ocean, but it represents thousands to tens of thousands of variations on this theme, not just one. SAR86, which had never been cultured before, is thus far the third largest taxa, but the data keeps going down and we can start to sort out the biology on the planet based on these categories of sequence versus what were thought to be individual reads. We can also do shotgun sequencing in the air. So we could set up sampling in this room, for example, and find some interesting things, but it's a lot more complicated because the lower density of organisms in the air, so we have to set up very high volumes of air, and we try to capture these in a liquid system, similar to any kind of cyclone vacuum. And it's easy with the time it takes to do large sampling to get contamination. So it's taken us a long time to work out these processes, but the machines have to be broken down and completely decontaminated very frequently so you don't get microbes growing in them. And then we just filter them the same way we do with seawater, and then do a shotgun sequencing of everything. And for our initial studies, we were sampling in New York City. This work was funded by the Sloan Foundation. And we found something that kept interfering with DNA sequences. We're trying to analyze what it was. And we finally treated the samples with a magnet, and we pulled out a tremendous amount of iron that's in the air in New York. And once we removed the iron from the samples, we no longer had interference with DNA sequencing. So in theory, there should be very little anemia in New York. It is not clear what the source of the iron is. There's a lot of construction, obviously, a lot of subway and train rails, but we don't know for sure what it is. We sampled at a variety of locations in downtown New York, one midtown office building that used to be occupied by a very now notorious financial firm that might explain some of the data you'll see in a minute. We sampled on the 22nd floor, both inside the building and outside. We sampled at a hospital in San Diego, inside and outside. We sampled at a, quote, typical home in San Diego and also from the end of Scripps Pier. And working with NASA, NASA is now providing us with all the HEPA filters from the International Space Station. So now what you know about the microbiome, you can imagine every time a new astronaut or cosmonaut goes into the space station, they're taking a couple 100 trillion microbes with them. And depending where they originated, you can have quite a set of diversity up there. And apparently the little tin can gets pretty rank at times. So we're going to be studying those microbes as well. So here's some of our findings. I don't know how clearly this is going to show up, but in the indoor air in New York, the majority of the cellular material that we got is human. And I guess that's not surprising. The statistic I gave out 2 nights ago is each of us shed about 500 million skin cells every day. When you see dust in places, that's you. That shows up obviously in the air inside this building. The second largest category was rodent cells and rodent DNA, bacteria, plants, other animals. When we go outdoors in New York City, the main cell material in the air is rodent DNA. And this isn't not a knock on New York City. I'm sure we'd find different species in different cities. But even at the level of the 22nd floor, 15% of all the material we can get out of filtering the air is human DNA. It turns out initially, because of all the famous rats in the subway of New York, we thought this might be mostly rat DNA. It turns out it's mostly mouse DNA. Probably if we got closer to the subway, we'd see a lot more rat DNA. All the rodent DNA inside this building, people thought was left over from this financial firm, but that's a different story. San Diego is obviously a much cleaner environment. The air inside and outside buildings is mostly microbial. You can see human DNA is 5 to 8%, but the hospital and the home look remarkably similar. When we look at the end of Scripps Pier, depending on what day it is and which way the wind is blowing, you see mostly insect DNA. You can see marine microbes, marine mammals, and because of all the surfers there, you still get a fair amount of human DNA. The big difference comes when we compare what microbes are in the air in the hospital versus the home. And it turns out it's not surprising people get sicker when they go to the hospital because the hospital air is loaded with virulent microbes, where the home was remarkably cleaner. So this gives us a different way to look at and measure the environment. And these methods are now getting to the point where they're becoming highly reliable. It would be interesting to do a global air genome, air quality comparison in cities. So as we look at this accumulating data, you know, that one of the questions, are we approaching saturation in our knowledge at all? At least our knowledge of gene sets. When you look at mammalian genes, it's pretty much saturated. If a new mammal is discovered, we might find 1 or 2 new genes, but we basically have all the same gene set for the most part, and most of those are known. If you look at any other branches of life, certainly microbes, smaller eukaryotes, viruses, we're completely in the linear phase of discovery. And it's actually easier to go out and make a discovery of millions of genes or thousands to tens of thousands of new organisms simply by using DNA sequencing than it ever has been before. So are these truly new genes, new gene families? And what we see when we look at this, it's a continuing expansion, obviously, of existing gene families, but I think the biggest surprise to all of us is we're continually discovering entirely new gene and protein families that have never been known before. And those of you who do genomics know that any genome from any species that's sequenced has a large number of unknown genes, and these are starting to accumulate in large families as well. The protein folding community is choosing some of these now to see if any of these represent new folds, or if it's the same set of folds, but a whole new families and functions. We're also applying genomic tools to stem cell genomics. It's very easy, just with a simple skin biopsy now, to take some skin cells, make a skin fibroblast cell line, and with a little bit of hormonal reprogramming, convert those into iPS cells, which can be programmed further into a range of stem cells. So one of the questions we had is how stable are these genetically? Stem cell scientists have been using pretty crude tools, but they've been looking at karyotypes of cells. Just seeing if all the chromosomes are there and if that looked reasonable. When we refine the level of discussion, there's tremendous numbers of reprogramming and genetic changes. This is true for embryonic stem cells as well as it is for iPS stem cells. It turns out cell-to-cell interactions in our species are very important for maintaining the genome in a stable fashion. And a variety of studies have shown that first DNA methylation is lost, and then you start to accumulate more and more changes in the genetic code. So we're doing complete genome sequencing at a variety of stages of stem cell development to see where these occur, can they be stabilized. A number of people have been investigating what are the cell surface constituents, such as sugars and lipids and the interactions to stabilize cells, and there's been some good progress there. But I think for effective stem cell therapy, unless we can find a way to totally block the changes, we're going to need to come up with ways to correct the errors in stem cells. And we have a variety of ways to do this now using synthetic genomic tools. So we can actually do genome repair on single stem cells. It's a question of what's the scale that has to be done at. Roger Laskin has automated the single cell sequencing that I mentioned to you earlier, and he's doing single cell analysis, genome analysis, and gene expression analysis on single stem cells. And with a variety of collaborators, is actually looking at the variation in genetic code and the variation in gene expression and regulation at the single cell level. So the tools are getting better all the time. Now, in 1995, we sequenced 2 genomes. After we did Haemophilus, we were looking for something to compare it to. And we thought it would be interesting to try and find the species with the smallest genome to try and get down to understanding life. As soon as you sequence a genome, and when nobody else had done this, and trying to annotate the genes and understand them, you find how important it is to have something to compare it to, and to compare it to in a meaningful fashion. So we contacted Clyde Hutchinson, who'd been characterizing this species, Mycoplasma genitalium, which has only 482 protein coding genes and 43 RNA genes. So we quickly sequenced that and started the first comparative genomic studies comparing it to Haemophilus. And we started asking a variety of new questions. How many genes are essential for life? What's the smallest number of genes necessary to run cellular machinery? And after trying a variety of experimental approaches of knocking out genes in various ways, we came to the question of, can we design and construct a minimal genome to answer this question? Because there's not enough selectable markers to do sequential gene knockouts to get down to a minimal one. And synthesis was the only way to proceed. As soon as we went in this direction, there were new questions. Would chemistry even allow us to make these large DNA molecules, because nobody had done it before? And even if we could we come up with a way to boot them up in a cell? We decided to start with Phi-X174, the small phage that kills E. coli, and the first DNA virus sequenced by Sanger and colleagues, one of which was Clyde Hutchinson in 1977. And we chose it for its historical purposes. It was also because it can tolerate very few changes in the genetic code, so we figured we'd have to get something highly accurate to get viable virus. Those of you who do DNA synthesis, you know that oligonucleotide synthesizers are very inaccurate, relatively inaccurate. It's an N-1 situation. The longer you make the oligo, the more errors you get. So we concentrated on new approaches for error correction. We synthesized the 5,386 base pairs and injected this into E. coli. And E. coli immediately recognized the synthetic piece of DNA as normal DNA and just started reading it, making mRNA, making proteins. The proteins self-assembled and formed the viral particles. The viral particles do what viruses do. They kill the host cell that made them, which is how we detect them. That's how we get these clear spots on a lawn of bacteria, and each spot has billions of viral particles. We call this a situation where the software is building its own hardware. The software was just the piece of synthetic DNA we put in the cell, and the hardware in this case being the protein-coded viral particle. But our goal was not to make a virus. Our goal was to make a complete bacterial genome, but now we knew we could at least make viral-size pieces accurately. So we thought maybe if we made 101 of these, we could try to find a way to assemble them together to make the chromosome. And that's what we set out to do. This was a long, laborious process, teams headed by Ham Smith and Clyde Hutchinson, where we divided this up amongst the labs and the facilities on the different coasts to start to put these pieces together to make larger and larger fragments. At each stage, to check the stability, we would clone them in E. coli, isolate the DNA, sequence it to see if it was accurate, then go on to the next stage of assembly. Once we got up to the pieces that were quarter genome molecules, 144,000 letters of genetic code, we started to reach the point where E. coli was unhappy with these really large pieces of DNA. And so we started looking around for other systems, and we found that brewer's yeast has a phenomenal ability to take in large pieces of DNA and actually assemble them together with homologous recombination. So the final stage in our synthetic genome assembly was to take these quarter molecules, put them into yeast with a small vector that contains an artificial yeast centromere. Yeast assembled these together, and we ended up with the first synthesized and assembled genome of Mycoplasma genitalium, almost 600,000 letters of genetic code. And at the time in 2008, this was the largest chemical molecule of a defined structure that was made. Fortunately, we had some really bright young people on the team, like Dan Gibson, who continued to perfect the methodology of genome assembly. Dan developed a single-step isothermal reaction that we now refer to as the Gibson assembly. And basically, it uses 3 enzymes. You just put the overlapping oligos into this one tube at 50 degrees for a while. and it ends up assembling very large pieces of DNA. This allows it to be automated. So we can go from the digital world to the analog world of DNA now without much in the way of human intervention. And also, well, we tested this by making the mouse mitochondrial genome very quickly. Now, New England Biolabs is selling the Gibson Master Assembly Mix, and those of you who prefer to assemble your own genes, you can do that now very cheaply and effectively. So we had two teams working. One was the chemistry team and the other biology team. So how do you boot up a chromosome? It's nowhere near as easy as it was with Phi-X174, which is very easy to get into E. coli. So we had a team headed by a French postdoc, Carole Lutigue, and John Glass, that developed methods over a couple of year period for first transplanting genomes. You can't pipette this DNA because it's too brittle. We have to move everything around and do all the manipulation in gel blocks and work out how to get that into a new cell. And that ended up with this study that we published in 2007, which I think is one of the most important of all the ones we've done that tell us something very clearly about biology and life. In this study, we simply swapped out the genome in a cell and completely converted one species into another. Because this is so central to what we do in synthetic genomics, let me walk you through this quickly. So what Carole and the team did was isolate the genome from M. mycoides, treat it harshly with proteases to remove any proteins, because at this time we wanted to know if simply naked DNA could be transplanted. We added a couple of gene cassettes to it so we could select for the chromosome, and also so it would turn cells bright blue. And we found ways to transplant that into capricolum, which is about 10 or 11% different than M. mycoides. So let me show you this movie that we have, very sophisticated. We moved the mycoides genome into the capricolum cell. And now we have a cell with two sets of genetic software. What happened is the same thing that happened with E. coli. The cellular machinery immediately started reading this new DNA, started making proteins coded for it. Some of those proteins were restriction enzymes that recognized the capricolum DNA as foreign material and chewed it up. So now we have the body of one cell and the genetic software of another. So what happened? In a very short period of time, we ended up with these bright blue cells. They're self-replicating. They've gone through billions of replications. And when we interrogate these cells, we sequenced all the proteins in the cell. There wasn't one single capricolum protein in the cell. When we checked with antigens, with antibody reactions, every level that we measured, there wasn't a single trace of the original phenotype. Everything in the cell was dictated by the new chromosome that we put into the cell. So we were feeling that we now had the systems to do this and knew that life is a DNA software system, and you change the software, you change the species. That's basically the fundamental basis of this whole new field of synthetic genomics. Now, we had a slight problem because some of you may have noticed we're assembling the prokaryotic chromosome in a eukaryotic cell. So to do the transplants with a synthetic genome, we had to find a way to get it out of the yeast cell to do the transplantation. So to work out how to do this, we developed a method for cloning entire bacterial chromosomes as eukaryotic chromosomes. The trick to this ended up being just adding the yeast centromere to the bacterial chromosomes, and now we have a wide range of bacterial genomes that we can maintain simply as eukaryotic chromosomes. And so this gave us the means now to try and isolate the mycoides genome from yeast and do the transplant. When we did this, it didn't work. This took our team roughly 2.5 years to completely solve. To make a very long story short, what we found is when we had initially isolated the genome from the mycoides cell, it was methylated. And that methylation protected it from the restriction enzyme systems in the capricolum cell. So to prove this, we isolated the 6 methylases from the mycoides genome, and we treated the chromosome isolated from yeast with the methylases, and we methylated it. And then we could take the methylated DNA, and the transplantations worked with the genome out of yeast. And to ultimately prove it, we removed the restriction enzyme systems from the capricolum cell. In that case, we could just take the naked DNA out of yeast and do the transplantation. So obviously, microbes use their restriction enzymes for protection, and it's not hard to understand why. If a whole new piece of DNA can come and just completely take over you as a species, protection from that obviously would be very useful. So we now had this new way to deal with things simply by adding a yeast centromere to a bacterial chromosome. We can now grow that bacterial cell as a eukaryotic chromosome. And those of you who work on bacteria know that most bacteria do not have genetic systems, and therefore most people don't work on a range of bacteria. They work on ones like E. coli that do have genetic systems. So as soon as we put the bacterial chromosome in yeast, we now have the repertoire of yeast genetic tools, such as homologous recombination, to make changes, isolate the chromosome, methylate it, do transplantations, and you can just work your way around this circle. So at this stage, we now thought we truly solved all the problems and could go on to try the synthetic chromosome. In fact, because the chemistry was working so well, we decided to synthesize the 1.1 million base pair and mycoides genome with some clearly defined changes in it. We started with 1 kb pieces. We put 10 of these synthetic pieces together to make 10 kb pieces. We put 10 of those together to make 100 kb pieces. And then we put 11 of those into yeast. Yeast assembled the 1.1 million base pair chromosome. We did the transplantation, and it didn't work. So we knew there had to be a problem somewhere along the way. Software engineers have debugging software to tell them where the problems are in their software code. So we had to create the biological equivalent of debugging software to find out where the errors were, and we did this by substituting various segments in the genome. We found that one of the 100 kb pieces was at fault. We had sequenced it initially with new sequencing technology, and it missed what we found with going back with Sanger sequencing, a single base pair deletion in the DNA A gene, which is an essential gene. So one letter being wrong in the genetic code out of 1.1 million base pairs made the difference between life and no life. We corrected that error, redid the transplantation, and we finally got the first synthetic cells. In fact, the first time we did this, we only got one single colony, but since then the efficiency of transplantation has greatly increased. We published this in Science in the middle of 2010. Now, one of the things we were most worried about is a control. You worry about things that could fool you into thinking you had synthetic life. So we developed early on, as you heard in the introduction, the notion of watermarking the genetic code. So you can clearly distinguish synthetic DNA from non-synthetic DNA. Even though we had a number of changes in the genome, spliced out a number of genes, had a number of points of variation, we wanted absolute proof. And also we think it's very important to mark synthetic genomes to distinguish them absolutely and be able to track them. In the first genome we synthesized, we just used the single letter amino acid code to try and write names of the scientists who did the work. but it was not very adequate. We were accused of not having very good imaginations. We didn't put in something like one small step for bacteria. So with this genome, the team went a whole lot further. A few of them developed a code that's in the first watermark that enables us to write the entire English language with punctuation and numbers using DNA code. And it's interesting, when we submitted the paper to Science, we got back one of the reviews written entirely in DNA code. And the reviewer argued that their code method was superior to ours. But in fact, it was basically just an ASCII code. And it was very important with biology to put in frequent stop codons. So we designed this code specifically for biological systems so that you don't create new peptides. The last thing you'd want to do, particularly in this country, is use a James Joyce quote and have it create a new toxin. So our code allows us to write things in DNA code without, with very frequent stop codons. As you heard, there's also a URL. This is the first species to have a computer as a parent, so it had to have its own web address for identity. And all we announced initially was that there was a code. As people solved the code, they sent an email to the URL in here, and after a number of people had solved it, we published the entire code and its interpretation, which included the names of the 46 scientists contributing to this over time, names of institutions. And I came up with 3 quotations from the literature that I thought was important. And the first clearly came out of Ireland with James Joyce. To live, to err, to fall, to triumph, to recreate life out of life. It seemed like a perfect quotation for what we were doing. The second is from American Prometheus, Oppenheimer's biography, see things not as they are, but as they might be. And the third, I chose Richard Feynman, what I cannot build, I cannot understand. Now, after this was widely published, we were surprised one day when we got a letter from James Joyce estate attorney wanting to know if we had permission to use this quotation. James Joyce was dead. We didn't know how to ask him. And also, under fair use laws, you can quote up to a paragraph of material without copyright permission. So we stopped worrying about that one. And then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. And we looked on the internet extensively, and this is the quotation that you'll find on the internet. And it turns out one of his early biographers made an error. And so for proof for what the original quote was, this individual sent us a photo of Feynman's blackboard when he wrote the original quote, which is actually a more interesting quotation, so it's unfortunate that it got distorted on the internet. And the quotation was, as you can read, what I cannot create, I do not understand. So we're changing the genetic codes that Feynman can rest in peace. And I think it is an important quotation because it's the essence of what we're trying to do with synthetic biology and synthetic genomics. Prove that we understand life by being able to build it and recreate it. So what are we doing with this? So we're trying to use the 60, now approaching 80 million genes discovered from these microbiome and ocean and environmental genomic projects. I view them as design components for the future, much in the same way the electronics industry in the 40s and 50s had resistors and capacitors, then ultimately transistors, then integrated circuits. Just a handful of components for everything we know in the electronics world. We have orders of magnitude more design components in biology. Part of the problem, though, is a large number of them are of unknown function. We actually have digital software at Synthetic Genomics for designing analog biological software, trying to design new cells for very specific purposes. We're in the process right now. We have sort of a design contest within the Venter Institute and SGI to come up with a genome completely designed within the computer. This is roughly a diagram of what's in the genome. We actually have 3 different versions that are being constructed right now, and we hope to know before the end of the summer whether any of our designs will live. But this shows we're trying to understand these processes. But if you look at this, the bottom line, unknown function is 21. Even in this design species, all we know is if we knock out one of those genes, cells die. We have no idea yet what they do, and this is the simplest cell on the planet. There's a nice study coming out in a week or so in Cell from this team at Stanford, actually modeling the mycoplasma cell in a very nice computer set of algorithms. And so this is the cover of Cell of what's coming out. It's very impressive, and 2 scientists from the Venter Institute participated in this. And this is a model of the mycoplasma cell where they're modeling all the different processes in the cell. And there's a little video that they gave me just to show you. This is obviously a sped up timetable. Looking at all the different process, what's being read in the chromosome, the DNA boxes as they're used, and you can start to see the rates of RNA processing, DNA synthesis at different stages. So this is just looking at a small piece of it. So it's taking the digital world and showing that we can successfully model, or at least this team can successfully model, a living cell in the computer and its functions, so we can track these different parameters. So it's a huge, complex study. Those of you who are interested in this, I urge you to look at Cell in the near future. So at Synthetic Genomics, we're trying to design new cells for specific purposes, capturing energy from sunlight, recycling CO2 into food, fuels, chemicals, etc. This year in Europe, the first genomic-based vaccine is coming on the market from Novartis. This is from a collaboration we did with them on designing a meningitis B vaccine from sequencing the genome almost 15 years ago. It works against all strains of meningitis. But we're trying to find ways to shorten this, and we're doing this with working on influenza. We lucked out for the most part with the H1N1 pandemic, although it killed a couple 100,000 young individuals. Didn't turn out as bad as predicted. And we're part of, my institute is funded by NIH to sequence influenza viruses from pigs, birds, and humans from samples around the world. And we're building algorithms to protect the rate of change of flu and also to synthesize it. So we're funded by NIH to make synthetic fragments of every flu vaccine. And right now, the collaboration we have with BARDA in the US government and Novartis has progressed very rapidly. So BARDA sends us an email, digital information of a new putative pandemic flu virus. My team's process is to synthesize this new virus in less than 24 hours. Right now we're doing it in less than 12 hours, and that still includes time for getting synthesized oligos from a manufacturer. So this is being shortened with adding in the oligosynthesis, and we'll have it down to well less than 8 or 10 hours very soon. We get this to Novartis, they rescue it and have it ready to go into scale up in their cell system. So we think we're pandemic ready, and we're actually working on now trying to get this approved for the annual flu virus. And we think we can actually predict what the virus will be before it occurs each year, And well before WHO even sits down to make its recommendation of what the vaccine should be for the year, we will have all the vaccines synthesized and ready to go. Now, the other night I speculated, because digital information can be converted into biological information. We do this now at the lab bench level, but we're trying to build a box. I'm calling it a digital biological converter. And maybe ultimately in the future, you'll just have a small box attached to your computer that you could download the vaccine from the internet in the case of a pandemic and have it in your own house. But obviously, there's a lot of processes before we need to go there. But digital biological information are becoming one and the same. It's faster and cheaper to synthesize a new gene than to wait for somebody to send it to you FedEx. It's now that way with viruses. When we synthesized the living cell genome, that was converting digital information into biological information. And digital information moves as electromagnetic waves about 95% of the speed of sound. So we can actually transmit biology at near the speed of sound and convert it back into biology. We're trying to use this with algae to develop new sources of omega-3s and new types of proteins, et cetera. And when you look at the way most omega-3s are, those of you take omega-3 capsules now, they're made by taking piles of dead fish and pressing them to get the oil out, which people refer to as fish oil, but in fact it's algae oil. And if you start with the algae, it doesn't smell fishy because it's non-oxidized like it is in dead fish. So we can change the process and use controlled fermentation versus fish kills to try and produce more of this. We're scaling up, as you heard, we've had a program with ExxonMobil to try and engineer algae to produce fuel molecules from carbon dioxide. This is a greenhouse we have in La Jolla, California, and testing different strains. And we just purchased an 81-acre facility out in the California desert to scale this up even more. But I don't want to raise hopes too quickly on this. This is still going to be a long-term process, because to make fuels economical doing this, it will be at least 10 years of large numbers of genetic changes that are required. Let me close just with actually where we started in this entire field, asking for a bioethical review before we did the first experimentation. We asked Art Kaplan's bioethics team at the University of Pennsylvania to take this on, and they took it on as a study and published this in Science in 1999 when we published one of our whole genome knockout studies. And they thought that we were taking very reasonable scientific approaches, and the only concern was the biological warfare concerns. The Sloan Foundation over the last several years has funded the Venter Institute along with MIT and the Center for Strategic and International Studies in Washington to look at the security aspects. And you can download these reports from jcvi.org. And when we announced our first synthetic cell in 2010, we got comments from the Vatican and the President of the US in the same day. And President Obama asked his new Bioethics Commission to take this on as their #1 challenge, and they did. And they published this excellent report in December of 2010, talking about what everybody in this field is finding new methods for biological containment as we have for laboratory E. coli, where it won't grow outside of the lab. So any new synthetic species couldn't possibly survive in the environment. Even if we grow it in large facilities, that'll have a chemical dependency, self-built-in suicide genes, et cetera, because we don't want to alter environments. We want to, other than by removing CO2 and reducing the amount of waste that we have. Obviously, I covered a lot of territory, but I'm delighted to be here, and thank you for your attention.
So thank you very much indeed for a wonderful lecture, and I told you he was a really interesting person. So now we have time for some questions. There are folks, the volunteers in the green t-shirts will be going around with roving microphones and a number. So if you put up your hand, please, then they'll come to you and I'll read out the number and that'll give us a sound. So right over here. Okay, so this is microphone number 1.
Rohit Talwar, Future. Thanks for an excellent presentation, Craig. What's your take on these services like 23andMe that offer to profile some part of your genome? What's your view on the value of those and the efficacy?
Well, I think 23andMe has an excellent website for educating people about genetics, and anybody that is contemplating having their genome done, I urge them to try something like a 23andMe service first, but to take all the results with a grain of salt. You know, these are gene chips measuring a tiny fraction of the genome, but I think they do an excellent job on education of relative knowledge associated with the test that they have, the relative value, are they validated by somebody else, in a good job of explaining to people. So you can get a pretty good education for a few $100s or whatever it costs. And if you find that interesting, then in the near future, you know, you should go on and get a complete genome done.
Very good. Number 3, please. A question, microphone number 3 at the back.
Yeah, just a question, Craig, on your call for a greater understanding of phenotypes so that we can match that properly with genomic data. Just a kind of a 2-part question. A, are you doing work in that direction or about to kind of boot up some sort of project in that area? And also, what is not adequate currently in initiatives like those, say, in Iceland or Estonia, that makes you feel that there's something missing?
Well, I think you can look around the world and around different research groups. There's some, for example, in the US doing phenomenal phenotypic characterization associated with heart disease. But there's no real attempt to integrate that with other information in other places. I think there's lots of early attempts to try and, in fact, do some type of phenotyping, but it's all at a pretty crude level, and there's no attempt to integrate a whole person. So we're having a lot of discussions, certainly at the University of California in San Diego, and the various institutions around there, whether we can start an integrated process of some kind there. We just recruited Richard Schurman to our institute, who's been working on a software approach that might aid in doing that. So we're at early stages of discussion. But while it didn't take an entire community to sequence the human genome like people thought it would, I think it will take the entire community to come up with a digitized, meaningful set of human phenotype information, because it's such a vast array of data types. You know, in the US, when you try and standardize medical care, surveys I've seen show on the order of 50% of physicians don't follow standard medical practice. So it's very hard to standardize things in medicine. It's very hard to even get agreement on what diagnosis is. And as studies show, as people delve in more and more into diseases associated, where you can separate them out using genomic or genetic tools, what people thought were single diseases are very much what people thought were like with a single cell of SAR11. It's a complete panoply of diseases, not necessarily even related to each other. So a phenotype can only be as good as the underlying definitions, and I think we're talking about the entire global medical community to try and get that. So it's not going to be trivial. The first step is getting people to recognize it's needed. People are turning away DNA sequencers almost in a vacuum, because that's what it is without the phenotype information.
Okay, we'll take another couple of questions. Number 1 first, that's down here, please.
Yes, after such a breathtaking series of studies, it may be greedy to ask one more. but about code expansion in the spirit of the Feynman remark and some discussion in the Nuremberg Symposium some years ago. To try and get 50 different types of amino acids synthesized with the synthetic genomes, you could have some codons that wouldn't be assigned, perhaps, but that would take, have to be careful about tRNA modifications or about expansion of code sizes, which would perhaps require alterations of the ribosomal RNA, the A and the P sites to optimize. But there's a whole field there.
Yeah, there's a lot of discussion about expanding the code. We're not lacking for diversity, but there are reasons to do it, and we've been trying to incorporate some of the non-natural amino acids, because if you do that into a genome, you can actually make a species that's viral resistant, at least at present. Somebody recently in a study showed if you go to a 4-base codon instead of a 3-base codon, obviously it gives you a huge expansion of the number of possible amino acids. I haven't seen a significant lack of diversity in the biological population with our 3-mere-base code, but there are some unique advantages to incorporating novel amino acids for making industrial organisms, for producing food or fuel. It'd be nice if you could make them so they wouldn't all be wiped out by a single viral infection, as this country knows well from its past with the potato famine. So whether we need to change the entire genetic code, I'm not so sure. Be curious to do.
Okay, 2 very quick questions, please, because we're out of time. At the back, number 3.
Yes, I'm Anneliese Kloet from Ghent University in Belgium, and I'm working in veterinary medicine, and I was wondering if you can create a synthetic genome for yeast. Can you do it for animals too in the future? Do you think we could create like a cow that gives more milk, or even create a synthetic human genome in the future?
I don't think anything is impossible, and it's always dangerous when people in science say things can't be done, because it motivates people like me to prove them wrong. I had a very interesting discussion a few years back. I was at a conference in Chicago, and a woman who was, let's say, extremely against evolutionary theory came up and attacked me and said, "Can you convert a human into a cow?" And I said, "Madam, what you're asking me to do is completely unethical." So we can definitely work with a single-cell species. I don't think it's going to be a huge leap to work with synthetic multi-cell organisms. Our problem right now is we're going to be adding another 1 billion humans to the planet in the next 11 years. I have no desire to make more of them. And cows actually are a huge problem. Making meat by using cows is probably the least sustainable practice we have. So not to put veterinarians out of business, Darwin, my dog, will always be happy to have a vet around, but we need to find good replacements for agriculture instead of just making better cows.
And the last question, number 4, please.
You mentioned accumulating mutations in growing stem cells. Is that more or less than in vivo stem cells? And thinking of your single-cell sequencing, have you found a lot of mutations over a lifetime in a person?
Well, the latter experiment really hasn't been done in a systematic way. My genome's probably been sequenced maybe 10,000 times now in different groups. Stem cells for this study, some of them were made from a skin biopsy from me, and doing a lot of sailing and surfing, I've been exposed to my share of UV light. And so the initial concern was this was just DNA damage from UV light that caused some of this. And My biggest fear was not that. It would be that in future studies you would have to take the biopsies from where the sun doesn't shine, which would be a much more painful process. So yes, our genome goes through constant change in life, and therefore this notion of having a single sequence, we will be sequencing our genomes in different ways multiple times, particularly with stem cell therapy. It turns out, as I said, the data seems to indicate the cell-to-cell interactions in vivo help stabilize the genome. And it's only when you take it out of that environment you start to get this deregulated set of changes. But I think it's very early on in that field to really know that for sure. One of the things that we're finding when we work at the single cell level, you start to see a pretty substantial level of stochastic variation. That's why even identical twins do not have identical fingerprints, because when we go from the 1 cell to 100 trillion cells, there's a lot of variance aside from just the genetic code outcome. So I think it's early on, but we have the tools now to start to answer some of those questions.
So with that, let me once again thank Dr. Craig Ventnor for a really fantastic lecture.
Thank you.